is Thursday, August 20th, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech. We're mapping the early stage signals before their big news. Aside from this one that's on the screen right now, which is obviously the the massive news of the
What
week.
is this meme even from? Like, what is happening in this? Is it a soccer game?
This is from F1, the movie F1.
Oh,
if you
okay.
haven't
Never
seen
saw it. Never saw it.
Phenomenal movie.
Interesting.
Brad Pitt, 60 in his 60s as an F1 driver, surprisingly quite good. But regardless, yeah, that's kind of where we'll start today. But obviously, Jeff, we can come back to this meme in just a couple minutes. If you want to start with the early stage biotech review.
Let me log in real quick.
Let's not show your password as your.
Oh, it's not going to show it. What do you think I am? Some kind of bad UI designer? We're good to go now.
Perfect. Excellent. Yeah, so that's where we'll start for now. But then in just a couple minutes, we're going to come back to why this meme is so relevant. But yes, right from the get go, Jeff, what's here at the top of the leaderboard this week for?
Yeah, so we had a weird like midsummer lull for the past couple of weeks, but it's been like a lot more things have been happening lately. And just forgive my stupid computer. We're just gonna lag out apparently. I gotta close a bunch of programs. I got PowerPoint open and Canva open, and we got all kinds of stuff going on. But yeah, we've got. I think Leal was here last time, but new top runner Kinexus. We're not going to cover them today, so I think that we should probably just like quickly go through what they did. $40 million Series A extension, totaling $110 million, led by Novartis. So congratulations. Yeah, so what exactly do they do? I didn't take a whole lot of time to look into this. It looks like it's neuro, though. Dude, okay, we're seeing a lot of this.
What's the breakthrough here over the last couple months?
Here's what I'm thinking. I think I mentioned it last time, too. Here's what I'm thinking. We know more about how to get drugs past the blood-brain barrier, and we know more about how these diseases work. That's got to be all that it is, right? Like, for whatever reason, we're seeing a bunch of pain medication stuff, and like neuropsychosis drugs, schizophrenia mainly, right? I didn't think that maybe this is where like my why it's confusing to me. I didn't think that schizophrenia would be that big of a market.
I guess
like how many people do you know who have schizophrenia
on the market? I think that's the big thing, right? There's such a huge unmet need, and so regardless of how prevalent it is, and it's relatively more prevalent than you might think when there's nothing on the market. That's a huge opportunity.
If anybody in the comments wants to sound off about why we're seeing a lot more schizophrenia drugs, I would be glad to receive that.
Yes, by all means. We can make this an interactive show. By all means, feel free to put a comment.
Yeah, you can comment.
Be part of the conversation for sure.
I will ignore you and then get mad if you make me sound stupid. So don't do that. Nice. Amygdala. Amygdala sounds like a Star Wars planet. Amygdala neurosciences. Okay, here's more neurosciences. Which one? What is this for? ALDH2. Can I guess what the indication is based on that? Ooh, I like this. Is cool. What's that for? Substance abuse. See, this is addiction. Like drugs for addiction. Who wants some drugs for addiction? Let me replace your current addiction.
Jeff immediately was able to pick out cocaine from the middle of a wall of text.
Orally available cocaine? I'm sorry.
Orally
I'm pretty sure it probably already is orally available, right?
Nasally available.
Yeah. Ooh, binge eating. I love that too. Yeah, this looks like a addiction medication play here.
There we go. Yeah.
Speak of the devil and he will show up.
If we just read in sequence instead of jumping.
Yeah. I'm not sure about you, but my brain just like scans, like look for words, and oh look at money. Ooh. So yeah, FDA cleared IND for ANS 858, ALDH2 inhibitor in substance abuse disorders. That's really interesting. A lot of like behavioral modification drugs, which is kind of like what GLP-1 is kind of, right?
Yeah.
Sort of, kind of, sort of.
Very true.
Yeah.
Nice.
And then anything else that's popping out? GaleMed. They had a new signal, Ben. So we talked about GaleMed last time because they did this. They had the 3 to 4 times cell death. They have the. You know what? I meant to make a LinkedIn post about this because it really is an interesting mechanism. Like what the GalMed is doing is they have a new like antibody drug conjugate class of therapeutics.
Right.
And instead of using an antibody, they use a bile acid molecule.
Right.
And they rely on the actual like biochemical activity of bile acid transporters to shuttle in the drug, which leads to way more drug in the cell because you can instead of binding being internalized, it's just being ingested by the bile acid transporter. And when the bile acid transporter does that, it doesn't leave the surface of the cell. When you have an ADC bind to a target, the whole thing goes down. So the more ADCs that bind to a cell, the less targets available. But with this, it just sucks it in and it stays there, and you can get more and more drug in the cell. And what did they do today? They had a signal, or yesterday, completes first in human feasibility study in 32 colorectal cancer. It's in colon cancer too.
Holy moly.
Which is, I think that their initial thing was not in cancer. Like they don't have NASH MASH, but now they have an active clinical trial, completed their first in human feasibility study in colon cancer. And 32 patients for a clinical trial, like for a phase 1, is actually nothing to,
Yeah.
That's pretty decent size. I'm wondering if you got to warn me, man. I got my headphones loud in here. I wonder if they had any kind of like activity readouts, no measurable failures, whatever that means.
Yeah.
What would a failure look like? Yeah, really interesting. I want to follow this company more. It's GalMed Pharmaceuticals. They're in Israel. Really cool technology there. But yeah, we should jump into the first story. What do you think?
Excellent. Well, before that, Jeff, we've got to come back to the meme of the day and the big story of the week, of course, right? For those of you who are not aware, Moderna's stock has rocketed up 150% on this news.
Dang,
so I just basically wanted to take a little bit of a moment to go back through why in the world is this story so big, and it really comes down to the fact that we have Keytruda that has become the most successful drug in human history, as far as I can tell.
Mm-hmm.
It was doing, Jeff. Last year, it did $25 billion.
Yeah, that's insane.
$25 billion over the course of the patent so far, the time where they've had that exclusivity, they've generated over $160 billion in revenue.
Yeah, and for those of you who don't know, Keytruda is PD-1 antibody used in cancer exclusively. I think what is the lead? Is it melanoma? Is like the lead indication that they use it in?
I think so.
But it's used. Like you look at clinicaltrials.gov, every like third clinical trial in oncology is like in combination with Keytruda. So it's used all over the place.
Exactly.
But it's a PD-1 antibody.
But anyway, why is this meme so important for the leadership over at Merck that's had this money printing machine?
Because of this.
Unfortunately for the Merck folks, at the end of 2027 or 2028, the patent on Keytruda expires.
Sad sounds.
Yeah. So they won't have that money printing machine anymore. So the question becomes, what can we do instead? So basically what they've been doing with Moderna is they've developed a cancer vaccine that can be used in combination with Keytruda, and it's significantly better than Keytruda by itself. So this is huge. As you have the one generic, this one money printing drug that's now going generic, it's like, well, sure, you can go with that. There's our man. Right on. Excellent. 20 some odd cancers.
Chris, we miss you. Okay.
We miss you, Chris.
Will you be at Meeting in the Mesa? Because we'll be in Arizona soon. Maybe we'll see you there.
One way or another, yes. We need to connect back up, Chris. Thank you. But yeah, so basically what's happening here is Merck is very, very happy because they've basically got an even better solution than Keytruda by itself that will be under patent for quite a long time and ideally continue to print money for them.
So this is their like mRNA vaccine, right?
Right. Yeah.
Okay, cool. I haven't been following the mRNA like personalized cancer vaccines a whole lot. I'm a little bit down on them, to be totally honest. I'm very curious to know if they work, because I don't think that they will. That's just me. But using them in combination with Keytruda will probably help them.
Why is that? Why do you think that's the case?
Why won't the mRNA vaccines work?
Yeah, for something like cancer.
Yeah, So I'm very skeptical of cancer vaccines as a whole, especially the mRNA-based cancer vaccines, because they just, in order for a cancer vaccine to work, it isn't like a prophylactic vaccine. A prophylactic vaccine, you're going to give a person, usually a baby, honestly, a vaccine, and then your body has time to create an immune response. And the actual like therapeutic immune response is the memory response, right? You don't give a baby who has measles the MMR vaccine. I mean, I guess you kind of would, but like you don't do that. That's not why you give the MMR vaccine. You give the MMR vaccine to protect them against measles, mumps, and rubella. So if they ever get exposed to that or chickenpox or whatever, then the memory response can actually enact the therapeutic effect, quote unquote. With cancer vaccines, you need to have that immediately, right? So you're actually relying on like the initial immune reaction, which is not really how vaccines are meant to work, right? So I think that they need to be more robust up front. You need to have like massive adjuvant activity or like some kind of like delivery mechanism to the lymph nodes or something to get more of like a punch, right?
Sure. But
and the mRNA ones, from what I can tell, and like the biology says, they work like slower, and they're a bit they can be a bit more robust. But I'm just, as a whole, cancer vaccines, both peptide based and nucleic acid based, they're gonna have problems.
Sure. But regardless, when combined with Keytruda, you're getting something that is better than Keytruda. So there's something to that.
Yeah, I mean, if you throw 2 bricks out a window, you're going to have more than one, right? So the question really, I guess, is like how much more therapeutic activity is being delivered from the vaccine versus just Keytruda by itself.
Right.
You know, I don't know. We'll see how it shakes out. Obviously, I don't work at Moderna for a reason, so they might know things that I don't know, but from what I can tell, the cancer vaccine space is going to have some significant innovation.
Fair enough. Cool. All right. Well, that being said, Jeff, on a very related topic, should we move on to a early stage signal that isn't, Merck and Moderna, that is very much related?
Yes.
Excellent.
So this is also, so Amplitude. What is it? Therapeutics. They're developing trans-amplifying RNA platforms for vaccines. So infectious disease vaccines, monoclonal antibody delivery, which I'm very interested in this, protein replacement, genetic medicines. It's a gene therapy play, right? So, but what this big thing here is that they deliver the RNA that encodes for the therapeutic. Could be an antigen for a vaccine, could be a full antibody, could be anything, a cytokine, a gene therapy, or hormone replacement therapies, right? They deliver that mRNA, and then a separate mRNA actually encodes for like a replicase, or what is it? A replicase. And what the replicase can do is actually amplify the mRNA of the therapeutic compound, right? Nice. So you've heard of self-amplifying RNAs.
I have.
A self-amplifying RNA encodes the therapeutic portion and the replicase portion on the same RNA, right?
Cool.
This separates them. So the replicase is being translated, and then it can just, which probably is better than the self-amplifying ones because it's shorter, maybe.
Is that a scientific term?
That's translation. That's the direct sound the ribosome makes when it binds. So yeah, what'll happen is the replicase is translated and it's made, and then bing, it binds to the mRNA of the therapeutic compound and just goes to town, making a bunch more of the mRNA itself. And then you can amplify the mRNA and then get more of the actual therapeutic after translation. So you can then reduce the dose because you're going to get more amplification inside of the body, and you're going to get a more robust, expression of whatever you're looking for, which obviously will be good, right? The big thing with them now is they signed this trans-amplifying RNA research collab with Lilly. So really good validation. Eli Lilly, I think it was like 3 or 4 months ago. Yes. Like 3 or 4 months ago, Eli Lilly was just going to town with their gene therapy stuff. A lot of deals with small companies like this for like gene therapy development. So looking forward to see how they use this. And again, it's just for like any kind of nucleic acid based therapeutic, gene therapy, hormone replacement therapy, things like that. So yeah, that's That's what they're doing, backed by Arch, Atla, NewPath, okay,
Nice.
Gates Foundation. So yeah, who is the leadership here? Corey Sago, Julie Sunderland, Jay Parrish. I think this name sounds familiar. Corey Sago. Why does it sound familiar?
Sorry, Chris. We'll miss you this year. Maybe we can pull some strings. Let's see what we can do here.
Oh, we can pull some strings. We have so many strings to pull.
Bold of us to assume we have a lot of strings.
Yeah, it really is.
Only the threads in our jackets.
Yeah, in the holes in my socks.
Yeah. Nice. Okay. Yeah, this is interesting. It's even with regards, right? You're talking about how this could be used inside of like antibody delivery. So this is somewhat similar to something that I worked on.
Hang on 1 second, Ben.
Yeah.
Didn't Eli Lilly just acquire an in vivo CAR T cell company recently?
I mean, dude,
they probably did.
Acquisition spree. They probably did.
I think I remember seeing like Cesare with Eli Lilly and some company, and all he posts about is in vivo CAR T cells. So it's got to be in vivo CAR T cells. What I can see happening here, though, is using this trans amplifying RNA platform for more robust in vivo like CAR expression for an in vivo CAR T cell platform. The Voyager description doesn't mention that anywhere, so like maybe there's not any kind of like publicly expressed interest in that. But I can definitely see this being used for that as well.
Right.
Yeah.
Okay. I'm just looking at. I think I found Cesare's post here. Eli Lilly acquired Orna Therapeutics for $2 billion, and Colonia Therapeutics for $3.25 billion.
So Orna is interesting. Orna does circular RNA, so I'm not sure that this could work with circular RNA. It might, and in fact, you know what? They might actually want it because Orna is circular. Because you wouldn't be able to do a self-amplifying RNA with a circular RNA, but you could do it with a trans-amplifying RNA. You had a circular RNA here, and then the replicase RNA here. Yeah. There you go.
Well, sound off in the chat, folks, if you have any thoughts on this, because, again, it's always more fun when it's not just me and Jeff going back and forth. Okay, all that being said, ladies and gentlemen, before we move on to the next story, we have a special guest coming at noon EST, so about 40 minutes from now. We're going to be talking with the CEO of Reprogram Bio, and they are turning cancer cells into allies. Interested to see what their approach is like. We had a somewhat similar company on a couple months ago now at this point. Asgard Therapeutics turning cancer cells into snitches. So we'll see.
That's my trademark, by the way, which I didn't know that Reprogram was like using the phrase turning cancer cells into allies when I said turning them into snitches. So, but yeah, it's such a cool. It's the gene therapies for cancer, really cool. This stuff is insane. Like, let's just turn the cancer cells into dendritic cells.
Yeah, absolutely.
Yeah, okay. That makes sense.
Love it. So yeah, that will be coming up in about 40 minutes. Lots of stories to get through in the meantime. Before we move on to the next story, let me tell you about Tetri Bio. It's all about who you know, but now you can catch prospects the moment they're ready. Deep company research, live signals from your socials, verified contacts, and a whole lot more. The BD engine built for biotech. That is Tetri.bio. Right on.
Based on the word Tetris for your perfect fit.
Yeah, this is something that Jeff and I are working on. It's our company, so feel free to go check it out. Tetri.bio. All right, so let's move on to the next story here, Jeff. I believe this first one is mine. Oh, you went to the Tetri website and everything.
Boom. No, I didn't. This is the ad on the Biotech Voyager.
There's the ad on the Biotech Voyager.
And you can connect your Tetri to Biotech Voyager, and you can add things to Tetri right from here. Wow.
Shakalaka. Yes.
Crazy.
So if you're in the BD area and you want to categorize all these companies that are at that early stage, you can do that right inside of the Biotech Voyager. Yes. Okay. Cool.
Okay, Ben, what do we got?
Yes. So let's move on here to AC Immune. So, Jeff, AC Immune. This is really cool because recently, if you will recall, we had the CEO of Base Immune, a company in the UK that is developing active immunotherapies for fibrosis.
Yeah.
in particular idiopathic pulmonary fibrosis, which is a massive mouthful. But anyway, I am increasingly very impressed by everything I'm kind of seeing inside of the active immunotherapy space. I think it's fair enough to say they're unique enough from vaccines. I think it's a little bit more elegant perhaps than a vaccine might be, where instead of just like developing like where the goal is you've got like some sort of infectious disease target that you're trying to get your immune system to memorize and go after. Instead, it's teaching your immune system, here is some sort of bad target inside of your body. Something has gone wrong internally, and so this is more defeating the enemy within. And so in particular, this company, if you scroll up to the top there, Jeff, and just kind of look. So instead of...
Do you want... This is... The formatting
like
is messed up.
Just like the picture, I guess.
Okay.
Yeah. Perfect. Yeah, so AC Immune is developing precision therapeutics against neurodegenerative diseases. They're going after Alzheimer's, Parkinson's, rare neuroorphan indications, and they're using their active immunotherapy platform to do this. And their lead asset is an anti-amyloid beta active immunotherapy designed to elicit antibodies clearing those neurotoxic amyloid plaques. So it's a really interesting approach, and the reason that they've kind of recently come back into the spotlight more recently is they've just received IND clearance from the FDA to begin U.S. clinical trials for one of its programs, and they've got fast track designation alongside it. So yeah, it's really cool to see what they've been doing in their sector of all of this.
They got fast track designation for it too. Oh,
yeah.
Okay.
Yeah.
For ACI
Oh,
7104.
it's the same one.
It's a different one, but I'm looking at...
Regardless, the company is, had a string of different successes here.
Yeah, I'm looking at this one. So it's interesting that they have like this active immunotherapy program platform, but like this asset is an NLRP3 inhibitor. I'm wondering why it's classifying it as an inhibitor. I'm going to pop into the press release really quick to see if they have any like interesting information that alludes to that. It's an orally available inhibitor of the NLRP3 inflammasome. So this seems to be like a small molecule.
Like a small molecule?
Yeah, they must be.
Active immunotherapies as well. Okay, so they're going into multiple different indications here. For a smaller company, like, hats off to you. Wow.
Interesting. So okay. And the idea, so they are doing the active immunotherapies. The idea and small molecules, but it's a morphomer. We don't know what this is. We know what the supra antigen platform is, but the morphomer, I don't know, man. But anyways, like when it comes to the active immunotherapies, right? The idea would be if you deliver one antibody for one epitope, like CD20, for example, that antibody binds to one epitope of CD20, right? It'll work, but if you can design a vaccine for, I'm not even super sure how these things really work. I should probably talk to the guys at Base Immune more to see how these things work. But you can design a therapeutic that will just like trigger your immune system to be making the therapeutics themselves, right? Which just leads to a more, I guess, robust, but really like the word that I want to use is like texturized response, right? Your body can just like do what it's good at and make the therapeutic itself versus you kind of like telling it what to use. Right. Yeah, I'm very interested in what this morphomer platform is. Yeah, could be anything. Could be anything. And of course, like the view of my monitor is just like insanely horrendous. Oh man. AC Immune. Precision medicine.
Probably their platforms if you go back up.
Yeah. Where's morphomer? It's like a transformer name. Super antigen, we know what that is. Where's morphomer?
That's going to be for their active
immunotherapies. Here we go. Accumulating knowledge has been used to focus the morphomer chemical library to approximately 10,000 CNS-optimized non-dye compounds, with characteristics that make this library an ideal starting point when targeting human CNS proteinopathies. Okay, so it's going to be, again, this is exactly what I said earlier. We're seeing a lot more neuro stuff coming out recently, probably because of the technology to identify and design blood-brain barrier penetrant therapeutics is just getting better, right? The BBB stops, like, I think it's like 98% of small molecules from crossing it. which is like obviously bad. How do you treat the disease in the CNS if you can't get the drug to the CNS?
Well, for the record, most of the time it is a good idea.
Oh yeah, it blocks from bacteria and all kinds of stuff getting in there, and you don't want like inflammation in your brain. So yeah, like I get it, but when you do want things to get in there, you need them to get in there, and we're just getting better at designing molecules that can cross it. That's probably like the majority of what we're seeing, to be honest. So, nice. Yeah.
Based in Switzerland, right on.
Swiss. All right, what else we got? Is there a cricket in your house?
I think there might be one outside. Can you hear that?
Yeah, I can hear it.
Can anyone else hear it?
That's hilarious. I'm looking around like, is there a cricket in my house or is it in my headphones?
That's hilarious.
Also, like, what time is it? Jeez, Mr. Cricket, go to bed. Okay. Anyways, what do we have up next? I think it's one of my signals.
Yes, it is.
Radio Farm Theranostics.
Yes.
Yeah. So Ben, do you know what the word theranostics means?
I can kind of throw a couple different pieces of it together, and I think it might be related to one of the stories later. Is it basically like this is a therapeutic that's used for diagnosis? And,
precisely. Yeah. So it's usually used
we have another one of those later on.
Okay. So you find it a lot in like the radioligand space, because radioligands can be flipped in and out. You can take the actual like payload you can flip it in and out.
Over, Jeff, like basically like just give me like a 30-second summary. Like, what is a theranostic?
Yeah, so theranostics are radioligand therapeutics or radioligand compounds that can be used simultaneously as therapeutics, and diagnostics, gnostics. So theranostics. And what you can have is a targeting agent, like this mouse, just imagine, that will bind to a certain target. Let's call it, I don't know, HER2, doesn't matter. And then you have the payload, my Aztec warrior arrowhead. Okay, you can attach this payload to this targeting molecule and it will go and it will kill things, right? But you can also attach this big green light to it, right? And it will bind to the same target and just show a big green light, right?
Nice.
So what you can do is you can use the same like compound to bind to the target and then visualize where the target is. Do we have good target expression, nice comprehensive expression across the tumor? Where are we going to be getting the actual thing to be binding? And then you can use that as the diagnostic and then take the compound, slap on the actual therapeutic, and now you know exactly where the therapeutic is going to land because you just imaged the diagnostic. So that's what theranostics is. And it's really only in like the radioligand space, and which is like mainly cancer. Right. So Radiopharm Theranostics just advanced to cohort 4. So they're pushing through phase 1 clinical trials I always want to know what the it's HER2. Okay. Breast cancer, HER2 positive breast cancer. I did not use HER2 as my example on purpose. I'm just a genius. Okay. That's the deal. I'm just a prophet. So yeah, I think it's a really cool use of radiotherapeutics. And I'm a huge nerd for like these new modalities. When I was making like my immuno-oncology video series, I did a whole thing on like the difference between alpha and beta emitters and like gamma emitters, which gamma is the diagnostic radio that you would put on, right? So yeah. Really, really cool. What do you think?
Nifty. So yeah, that's going to be somewhat related to a story we have right before our conversation with the CEO of Reprogram Bio at noon.
Nice.
That just got 2 different FDA designations, and it's coming and it's in a very similar space, but for kidney cancer instead. Yeah. So we'll get to that in just a bit. But yeah, no, this is super cool. So is this like a relatively new thing? Is this something that's been around for a long time? How long have these theranostics been a thing? I'm just not familiar with them.
I think the imaging diagnostic piece has been around for a while. What's happening now in the radiotherapeutic space is like the discovery and optimization. Well, really, it's the optimization of the infrastructure. Because like when you make it isn't a new thing, especially radiation therapy. It's been around for over 100 years. But the idea of or being able to make reliably a radio ligand therapeutic, it's a pain. It's a pain because the things are decaying. Like you've got to get,
They
you've
quite
got to
literally have a shelf life, regardless of how hard
Yeah.
you freeze them.
yeah, like it's yeah, like with an actively decaying like radio isotope, it's just a big mess. And the thing is like, uh, if you have a long half life on your radio isotope, that's good for storage and good for travel, right? Like you got to make it somewhere, give it to put it on a plane and get it to the patients. If it's decaying slowly, then it isn't super dangerous and you can travel with it and it's not going to decay before it gets to the patient. But then once you inject the thing into the patient, if it isn't decaying fast, which is like what actually causes the therapeutic effect, you don't get a lot of therapeutic effect. So you need to have the thing stick around for a long time, and it's a big pain in the butt in the body, right? If, on the other hand, it decays really rapidly, right? Well, now it will work really quickly and well in the patient's body, but you can't travel
For
with the
3
thing.
hours.
It's gonna exactly like if the half-life is 6 hours, you got to make it in the hospital, right? Which a lot of people are doing now. So the big innovation is these cyclotrons, I think they're called, where you can actually like make the radio isotope like on site and then manufacture the actual radio ligand therapeutic. Like I think like literally in the hospital, or at least like nearby. So like look into like alpha actinium stuff, and I think the big one is like lead. I don't know. I knew more about it like 3 months ago, but there's a bunch of different like isotopes that you can use, like alpha and beta emitters.
Astatine, right?
Astatine. Yeah. What did I say? I don't know what I said.
You said
actinium,
lead, which you're definitely not using as.
Yeah, it's astatine. Astatine 211, I think.
The everything from. Them.
Oh no, there's definitely lead, like lead 116s or whatever that are
Really?
radio. Yeah, they're radioactive.
Interesting.
Yeah.
Cool.
So, but yeah, the radio theranostics space, really cool.
Oh, they're from Dananda.
Victoria. Yeah, we got some friends in Australia. Biocena, shout out.
Yeah. Speaking of Australia,
anything else?
Did you see the video? Remember that guy who vibe coded a cancer vaccine for his dog?
Yeah, with ChatGPT.
Yeah. He is now part of the next Y Combinator batch, and he's
No way.
building it into a company.
Nice.
Yeah.
We live in a different era, dude.
Yeah.
Different era.
There's people who still didn't know about that. I was talking with a guy yesterday, like one of the big, like someone who's been in cell and gene therapy, pretty much as long as we have been alive, and he hadn't heard about this cancer vaccine story. And so he's like, I got to go check that out. It's like wild. We just kind of assume, right? You see things on LinkedIn and you're like, okay, everybody else knows about it now too. But there's just so much cool stuff that's coming out that people are just missing. Like a vibe-coded cancer vaccine. Who would have thought? So, yeah.
And then it worked, which like what?
Yeah.
Weird.
It's a different era, man.
It is.
Era. Faux show. Excellent. Well, ladies and gentlemen, we will be having our special guest on in about 25 minutes, the CEO of Reprogram Bio. He's turning cancer cells into allies, so really looking forward to that. In the meantime, we've got some pretty interesting signals to get through. And so if at any point you want to be part of the conversation, feel free to sound off in the chat, and feel free to throw in your 2 cents. We would be glad to turn them into $2. I don't know why I said that. So the next story here is going to be Calavir Therapeutics, Jeff. You want to look them up?
Yeah. It's oncolytic viruses.
Therapeutics. Yes. So this one's mine. This one's interesting because very recently there has been a kind of a little bit of a resurgence inside of the oncolytic virus space. There historically has only ever been one FDA approval of an oncolytic virus.
Mm-hmm.
That was back in the early 2010s. I think it was like 11 years ago at this point, so mid 2010s. And very recently, there was a second FDA-approved oncolytic virus therapy that was, I think it was earlier this month, that got that got approved. So big signal. And so here is kind of another oncolytic virus therapy that's up and coming. That's potentially going to be able to move forward. They just raised $14 million as a Series A extension, and they're going after. I'm looking here at just quickly which therapeutic they're going after. Does it say there what they're going after?
Yeah, it's head and neck, and
head
then
neck cancer?
microsatellite stable colon cancer.
Okay, right on. So yeah, really interesting to see potentially how this space might really be primed for some sort of resurgence, because yeah, there's been a lot of improvements made in terms of like the engineering of oncolytic viruses as well. My understanding is that historically oncolytic viruses very much just kind of focused on get inside of the cancer cells, grow like crazy, burst them apart, and kill the cancer cells, and that's kind of the end of the job. And there have been some really interesting additional payloads that are starting to get added into these oncolytic viruses that are really designed to not only destroy cancer cells, but really prompt the immune system to get engaged and to form memory and to go elsewhere in the body to find other potential cancer tumors as well in the same indication. So yeah, this space might be ripe for a little bit of a comeback. Thoughts, Jeff?
Yeah, for whatever reason. So the actual like innovation probably isn't actually the increased number of payloads. I've been seeing a lot of like oncolytic viruses that deliver, they'll lyse the cancer cells, but they also deliver IL-2, right? Or what have you, even like bispecific antibodies. I just don't really know. Like it operates under the premise that if we want to turn the hot tumor or the cold tumor into a hot tumor, all we have to do is pump in some IL-12 and maybe inhibit TGF-beta, right? I don't really know it's that simple. And I guess also one of the big problems is that oncolytic viruses, from what I understand, you need to have access to at least a large-sized tumor, because it actually relies on the tumor itself creating more of the therapeutic, because these viruses are actually competent, like replication competent viruses. So they get in there and they'll infect some of the cancer cells and they'll make a bunch more of themselves. And then you effectively have increased the dose of the virus, and it will go into the blood and it will have an abscopal effect around the body.
Fair.
I forget what I heard, but there's problems with that. There's problems with like that approach. Trying to get them into like a big enough primary tumor to get an effective dose. So I don't really know what the actual innovation has been. Maybe you're right. Maybe it is just like, well, we can increase the genetic payload in the viruses, right? But from what I understand, I think it's probably more of like a delivery and like a preferential infection and preferential expression in the cancer cells thing. Like, do you know the company Trogenics?
We've talked about them before.
Yeah, like we're getting a lot of things like this. So like Trogenics, it's based on. Did you see The Odyssey yet?
I have not.
The movie The Odyssey. So it's based on like it's called like Trogenics as the Trojan horse, right? And their platform is Odysseus, the Odysseus platform. Really, and what they do is it's a virus that's used to treat brain cancer, and they rely on like super enhancer elements that are primarily active in like glioblastoma cells. Right, so they'll have like the expression of their therapeutic payloads driven by super enhancer elements that are only like acted upon inside of a specific cancer cell subtype or whatever. Right, this is a lot of what I'm seeing as far as innovation goes.
Can I pause you for just a second?
Mm-hmm.
Is it a little bit of a misnomer to want to call your precision oncology platform Odysseus? Because kind of the whole point of the Odyssey is that he takes way too long to get home.
Sure. You get the idea, though. It's the Trojan horse. Come on, man. But yes.
It's like
there may be some
confusion
confusion
there. Specific direction, but it's going to take 10 years.
Yeah. But this is, so they have these like synthetic super enhancer elements that are primarily like acted on in aggressive cancer cells, basically, right? And they'll use them, I think it's in glioblastoma, definitely in like brain cancers. So I think that like this, and then in terms of like the infection, the preferential infection of cancer cells, there might be innovations happening there too. Right? Because you do need to have like selective expression or a selective infection in the cancer cells, and or at least robust selective expression in cancer cells for any of this to be even remotely safe.
Agreed.
Right. So, but yeah, I like the idea. This is like my Dispatch Bio like itch arises here, because the whole, I don't know, man. I don't want to get into it again about Dispatch Bio, but I really do feel like it's an oncolytic virus with more moving parts. So, anyways, that is pretty cool. What was the signal again? Sorry.
They've raised an additional $14 million through Series A extension.
Nice.
Yeah.
Nice.
So again, like I just, I always like to cheer for the underdog. Oncolytic viruses have definitely kind of been a little bit slept on. They've had a bit of a rough season, and it might be that we're now kind of primed to see some nice results on the heels of a very recent approval, the second inside of the oncolytic virus space.
Yeah.
In total from the FDA. There have been other ones that have been approved in other parts of the world, but inside of the FDA, there's only been 2, and the second one was like less than a month ago.
Mm-hmm. Huh.
Yeah. Shout out to
oncolytic
Kyle
viruses.
Here. Hopefully we're saying your name correctly.
I think we are. All right. Shall we move on?
Yes,
Tolerance
indeed.
Bio.
Yes.
Ben, we met these guys in Philadelphia. Tolerance.
We did. Yes.
They're doing like thymus therapeutics, but when I saw this signal this morning, I was like, what is going on with these people? So they acquired an IL-7 therapeutic, a phase 2 IL-7 therapeutic. Long-acting IL, yeah, weird. So good luck pronouncing that. Let's give it a shot. Affinaptekin alpha to restore yeah, to restore T cell function and thymic activity. Okay, so it is still a thymic play. Ben, what do you know about the thymus?
The thymus. Thy must educate me.
Okay, it's right here. So it's the master regulator of immune tolerance. It basically is what helps you not die from your immune system. And in autoimmunity, it obviously has some issues because we have autoreactive cells. So I think, from what I can recall, Tolerance Bio has got a bunch of different platforms. Their main one that I was kind of talking to them about was their iPSC-derived thymus epithelial cell, like therapeutic. It's a thymus cell therapy, right? And you can implant it like in your arm, right? And just have like a mini little thymus like satellites kind of all over the place. And you can prime the thymus cells to select against the autoreactive cells, right? So you can have like a second thymic selection process, right? And you can eliminate autoreactive B cells or T cells.
Cool.
IL-7, though, this implies that thymic dysfunction is at least driven by or can be restored by IL-7 activity. Thymic dysfunction is driven by a loss of IL-7, and then you can restore it by restoring IL-7 activity. But I don't know. I'm not a thymus expert. I don't really know what IL-7 is doing there, but I thought that it was really interesting that they went from private, preclinical, to public. I think this was like they got like some kind of public thing. They're like on the NASDAQ now, I think. And it's phase 2. It's phase 2 asset. So they're now a clinical company. So let's see. So exclusive license agreement with Neoimmune Tech, which is on the so is this Korean, the Korean NASDAQ, for the development and commercialization of their NTI-7 IL-7 fusion protein for certain thymus-related indications in America and Europe. Yeah. So maybe they're not public, but they're going to initiate the phase 2 clinical trial. So like good for Tolerance. Again, we met them in Philadelphia. Really smart guys.
I did a video with them, right?
You did do a video with them.
Yeah.
Yeah.
Let me see if I can find it and I'll throw it in the chat.
Yeah. So really cool stuff. The it's a totally new thing. So the thing about autoimmune therapeutics, especially the new ones, right? Like everyone goes after the B cells because of the autoantibodies, right? Autoantibodies are causing inflammation. That's basically the entire premise, right? But there's T cells also. T cells can be antigen specific. And a lot of like the therapeutics kind of ignore the T cell population to a certain extent. This just
Okay.
eliminates them both.
Just put it in the chat, folks. If you want to go check out the video I did with Tolerance Bio, if you want to dive into the story a little bit more, there you go. Sorry, it's a terrible link.
Oh, it's on LinkedIn?
Yes.
Nice.
Of course.
Where else would it be?
Where else would it be?
So yeah, I have no idea why the Voyager made this their picture. I guess that's supposed to be a thymus. I think it actually is supposed to be a thymus. Interesting.
A thymus in outer space.
So yeah, I think it's a really interesting approach to autoimmune disorders. Just like recreate a autoreactive cell specific thymus structure in your arm. Really, really cool. Based in Philadelphia. I think they're working out of B Labs. I could be wrong there, but I think they're working out of B Labs in Philadelphia.
Possibly. Shout out
Don't
B
know.
Labs, friend of the show.
Yeah, shout out Brian Kennedy.
Yeah.
So yeah, any thoughts on this?
Yeah, no, it's nifty, and always good to see a cell therapy play.
Yeah, with thymus cell therapy play. That's really interesting.
That's
So-
got to be fairly rare.
Yeah. And yeah, there's definitely a couple other people who are doing it. They aren't alone, Tolerance Bio, but it's very rare. Very, very rare. But yeah, that's all that I had on that. It's, I mean, I didn't know that IL-7 they were going to use it for thymic disorders, right? Like, autoimmune or whatever. I thought that it was like a completely different thing. But apparently IL-7 has got some activity in the thymus.
Sweet.
So, yeah. Also, let's do this really quickly here. I want to see like if I can get some this Philadelphia. Do we have them in here? That's New York. My geographical illiterateness is coming out here.
I mean, you're
There we
talking
go.
to a Canadian man.
Tolerance Dispatch 4, Gemma. Remember Gemma?
Gemma.
Great-
Shout out
great
Gemma,
company.
friend of the show.
Yeah.
I did a video with them as well.
You did.
I think you shot it.
I did shoot it. Yeah, you're right. You are right. I'm an excellent cameraman, aren't I?
You are.
All right, what else we got, Ben? We have 10 minutes here.
Yeah. Maybe
Before,
Before, yes, in 12 minutes or so, we will be joined by the CEO of Reprogram Bio, who is focusing on turning cancer cells into allies. So, ladies and gentlemen, stick around for that, coming up in 12 minutes. You can get your questions in now. If you drop a question in the chat that you have for him, you can put it there now, and we'll get to it in a prioritized sense versus, if you all are piling in once he joins and we're just inundated by the many questions from all 8 of you. It might take a little bit longer.
yeah, let's do it. What else? What's the last signal, Ben?
All right, last
Norway?
signal here is a drum roll, please, because
Oh, it's more radio pharma stuff.
Norway gets breakthrough plus fast track from the FDA. So this company is very much what we were just talking about, Jeff. They're also kind of a theranostic play, but a bit more on like this diagnosis side. They've developed some sort of interesting application that allows them to image and track and diagnose cancer in kidneys really, really well. And apparently that's big enough of an unmet need that it's resulted in them getting not one but 2 different designations from the FDA, both breakthrough and fast track. Yeah, the key thing here, basically, yeah, they develop targeted radiopharmaceuticals, automated synthesis systems, and GMP manufacturing modules. So they're kind of doing everything under kind of this whole diagnostic and therapeutic arm. And they're going after solid tumors, hard to treat ones, in particular ones that are found in the kidney. And their lead asset is a gallium-68 PET imaging agent that targets carbonic anhydrase.
Yeah, that's PET imaging.
Yeah.
Yeah, so this is mainly like a diagnostic company then. Do they have any therapeutics? I see that they have therapeutic need. You could do it. I mean, you just pop in a different radio ligand or a radioisotope. Yeah, I don't know a lot about carbonic anhydrase. Is that IX or 9? I've seen this around, but like I'm not an expert here. I know that like when it comes to like kidney cancer, is it hard to diagnose? I don't know. I was like a bone cancer guy, right? Like I don't know how hard I mean, I would imagine that it would be more difficult than like skin or maybe bone, but yeah, interesting. I also have never heard of gallium-68. This is a new one too. And a lot of the imaging ones are kind of like you don't see them a lot.
Right.
At least like in the therapeutic space. Yeah. With therapeutics, you have like there's like 3 or 4 you always see. But gallium-68, I'm sure that it's like popular. People are probably like thinking, what an idiot this guy is.
Yeah, if anyone in the comments knows a little bit more about this, feel free to sound off. But yeah, really want to just highlight this one, given that they got both breakthrough and fast track from the FDA on the same day. So yeah,
Yeah, so I think that this right here, like 177 Lu, this is the therapeutic one. This is lutetium.
Okay.
Yeah, lutetium. So they do have like a gallium, lutetium. This is a theranostic right here. This is a theranostic. So this is the diagnostic radioisotope, and then here's the therapeutic isotope. This is a beta emitter. And then this must be like whatever the targeting molecule is, NYM, whatever. This targets the cakes.
Can you do this at the same time then? Is this kind of what I've been missing? Like with a theranostic, not only can you also basically hit at the cancer while tracking it, but it also allows you to.
No, you do it sequentially. You do the diagnostic first,
okay,
and then you probably do the diagnostic afterwards as well to see if you've gotten any reduction. Yeah.
Oh, Asgard Therapeutic is also reprogramming cells as well using an AAV vector. I believe actually they're using an adenoviral vector. We've had Asgard Therapeutic on the show before, so if you go back to the YouTube channel, you should find their interview. But yeah, they are super cool. So what is the main difference between this company and what are the commonalities? We're going to find out.
Yes, we're going to find out in about 5 minutes.
This is a great question. Thank you for getting it in now. Shifu999. Love it. Yes. Yeah. So yeah, we'll do that one first. Hopefully they don't mind a direct comparison between.
Oh, they have to know. With this unique of a therapeutic approach, they have to know every single person who's doing this. And like Asgard's is basically, it seems to be very, very similar. I'm gonna guess that there's probably differences in the genetic, the combination of like transcription factors that they use.
Right.
And I'm not super sure if reprogram is like turning the cells into dendritic cells specifically. It might be a different kind of APC. I don't really know. But yeah, I'm gonna guess it's dendritic cells, maybe some macrophages. I have no idea. So yeah, we're gonna find out. I'm gonna guess that it has to do with a difference in the actual like assortment of factors that they use. It has to be. You couldn't use the exact same ones, but it is very similar nonetheless.
Nice. Okay, Jeff, we've got a couple minutes here before the interview starts. So maybe we could go back just to the leaderboard and see who else is rounding out the top 10 there.
Yeah. So what if we went to a subcategory of let's try and find these.
Folks, if there's one particular company you would like us to look at and provide commentary on, sound off and let us know. And if you're not subscribed to the Biotech Voyager yet, please do on YouTube, on X, on LinkedIn.
So here we're going to find any gene editing or gene therapy company in oncology that has had recent signals. There's 14 of them. Who do we got? Dispatch Bio. What's the first one? Dispatch, Obsidian, Umoja. We had CoRegen on. Why does it consider them to be a gene and gene therapy? That makes everything else makes sense here, but like CoRegen.
Shadow CoRegen.
Oh, because it's a gene editing. They have a Circ3 knockout. Yeah, that makes sense. Ben, what do you know of Avencell?
I don't know much. Let's take a look at them.
So they have a really cool technology. It's a universal CAR T cell. And whenever you hear like universal cell, you think of like it's not going to get rejected. It's a universal cell. You can just use it in everybody. But when it comes to CAR T cells, I guess switchable is like the preferred industry term. I usually use universal. It's a CAR T cell, okay? But usually with a CAR T cell, you have like the CAR binds to the target. Like here's my CAR binding to the target. My fingers in this scenario are that antibody fragment. That's like covalently bound to the CAR receptor, which is my arm, right? What Avencell and other companies are doing and I think that Arcellx has one too, but they just got acquired by Gilead for like the moon. They make a CAR receptor that's like a Captain Hook nub, right? And then this thing itself, the receptor itself, can't bind to anything besides an adapter. Bink, right? So it binds to an adapter, and then the adapter is specific for an antigen, which means you can have one cell. You make one cell population, right, that expresses the universal CAR receptor, and then you just manufacture dink. Here you go. Dink. Different adapters for different antigens, right? Which is significantly easier than making an entire new cell, right? So you can have one cell population, and then what you can also do is you can co-infuse different adapters to do co-antigen, like multi-antigen targeting. And you can also monitor, or I guess control, the activity of the cells by changing the dose of the adapter.
Cool.
Right? So like super cool stuff. I'm not sure, their signal was kind of a long time ago. It's yeah, why is it not here? They definitely have like a signal recently. But yeah, no, I always, whenever I see any of those companies, Umoja had one for a long time too. Not super sure where it went. They mainly do in vivo CAR T cell stuff now, but like Umoja had one. Scepter Bio, random in Australia, they have one. There's Senti. Remember Senti, dude? What a great company. Oh, you there? Nope, you're muted.
I'm just gonna email Rustum quick.
Oh, okay, I lost you for a second, and I was like, oh no, I'm alone. Siren, shout out Nicole. Yeah, I mean, I could go into my. I really want to know what people think about Dispatch Bio. I've got some questions, and I would love to talk to. I forget who you had a conversation with, Lex Johnson, I think it was. Yeah, I really have some questions about Dispatch Bio. And the reason that I'm so intrigued is because I have to be so wrong about this, because like the Parker Institute for Cancer Immunotherapy, they're involved. Carl June's involved. Like everyone from UPenn's involved. And here I am like, that's a stupid idea. Like there's no way that's going to work, right? So I've got to be wrong on this. So I'm really looking forward to trying to get him on the show. Anyways, I heard a bing.
You did. It looks like Rustum has joined the green room, so it is the pretty much the top of the hour here, Jeff. Should we bring him on the show? And folks, if you have any questions for him, get them in early before everyone else piles in. Yes, so very excited to welcome Rustum to the show. With no further ado, let's bring him on. Oh, no. There we go. Hey, there we go.
Hey, guys.
Rustum, welcome to the show.
Thanks for having me.
Excellent. Well, yes, I guess before we get started, we've got loads of questions we want to ask you, but can you just give us a little bit of a background? What's kind of brought you along your entire career to reprogram?
Sure. Well, first of all, thanks for having me, guys. Ben, I'm actually surprised you're already back to work after a recent addition to your family. So
Ben took approximately 14 minutes off, and then he was back at
it. It does help that my office is this studio for now anyway is still in my bedroom, so my wife is still supported. I'm not 45 minutes away.
Yeah, Ben is actively rocking his baby with his left hand.
Multitasking, right? You do what you got to do.
Mm-hmm.
But yeah, thanks again for having me. I'm Rustam, co-founder and CEO of Reprogram Biosciences. And yeah, I'm a scientist by training, did my PhD in academia, did a short postdoc, and then moved to the Bay Area, where I worked in 3 different startups before joining GSK, where I spent about 4 years. So throughout my career, I used reprogramming as first to model the disease. So reprogramming, for those of you who are not familiar, is essentially changing what a cell type is, and you can do that in a dish. And now we're trying to apply the same concept in vivo for therapeutic purposes. So in the beginning of my career, I was basically making motor neurons from patient samples from ALS in order to understand the disease and how the disease, how we can target the disease for therapy. And then as I moved to industry, I was using these neuronal models and muscular models to screen drugs to try to understand what kind of drug we can develop. And then throughout that experience, I realized the potential of reprogramming, and I was kind of surprised that no one has applied it in vivo for therapy, and decided to start reprogram last year.
So can I have a question on that too. I was also surprised. And I'll be honest, the first time that I kind of came across this was through a company called Asgard Therapeutics. But you guys are doing something very similar. And my first question when I saw Asgard was, wait a minute, no one's done this yet? Like, so I guess my question is, what exactly is the innovation? Or is it just, we had a good idea and nobody's tried it yet?
Yeah, so there's both innovation, and also I think there's a reason why no one has tried it yet. But I feel like, so let's start on the innovation part, right? I think reprogramming as a whole, there's like, we just scratched the tip of the iceberg in terms of identifying the ways of how to transform one cell type to another, or activate one cell function in a cell, right? And one of the biggest reasons for that is we had no idea of what the cell type is, right? We didn't have an accurate understanding of what a cell type would look like until single cell sequencing came along. And now we have, this vast majority of data that we can mine and we can use to even do these reprogramming simulations in silico. And that's what we're doing at our company. And then the second piece to that is delivery, right? How do you actually deliver genetic material that encodes for your reprogramming factors in the right cell type in the body? And over the past decade, there's been emergence in delivery modalities, most notably mRNA, which we are using for our lead program. So I think the convergence of these two developments makes reprogramming therapeutics possible now.
Yeah, that makes sense. So I guess for the audience, could you just kind of give us the 45-second overview of what the therapeutic actually is?
Yeah, so reprogramming therapeutics is essentially genetic material that you deliver inside the in vivo in patients. Once it goes to the right cell type, it initiates a cascade of changes that forces that cell type to behave like a different cell type, or it activates a function within that cell type for therapeutic purposes. So that's in a nutshell is what we call reprogramming.
Right, and the underlying like biological foundation, like the very foundation, is that every single cell in our body has the genetic capacity to become any cell in our body.
Exactly.
Right? So every single cell is genetically identical, give or take some mutations. But every, my eyeball cell can become a renal cell, right?
That's correct.
It's just that it has the right reprogramming factors in my eyeball cell to make it an eyeball cell, differentiation. And you are massaging the differentiation cascades to make whatever cell you want into whatever cell you want.
Exactly. Yeah. The analogy that I like to use is an orchestra, right? Orchestra has different instruments, but depending on what the instructions of the conductor, you can perform different melodies. So in this instance, instruments are your genes, right? There are 20,000 genes in a human cell, but not all of them are active at the same time. So the activity, the gene expression, mRNA, is, let's say, the music, right? The piano plays, or the other instruments, drums. So the conductor in this case is master regulator, what we call transcription factors. And those are the special genes. There are around 1,700 of them in a human genome that can selectively turn on different pieces of your genome. And we use those for reprogramming.
Okay. Ben?
Yeah, a couple questions. So first of all, we do have one question we want to get to fairly quickly. Just want to kind of get through some of the standards here first. So what is the main indication that you're going after? What's the main disease application here?
Yeah, so our lead candidate is we're developing it for solid tumors. Right now, we're testing it in colorectal and non-small cell lung cancer.
Excellent. And yeah, like with when we just kind of paint the broader picture here, right? Just to make sure we're kind of all on the same page here. Why is it that most therapeutic applications for solid tumors haven't worked well in the past?
Yeah, so I think what tumors do very well is that they hide. They hide well from the immune system. And to be frank, there have been very good developments in the solid tumor space, but I feel like we can kind of we can try a different approach. And in our case, rather than attacking the cancer cells directly or trying to kill it with the therapeutic, we're utilizing some sort of a unique approach where we're not trying to kill cancer cells. We're actually trying them to turn them into allies, right? To turn them on our side. And that's what tumors, like cancer cells, they do already to our body, right? They recruit, they make your body build vessels to bring nutrients. They transform your immune cells into allies on their side, so they can hide from the immune system better. So we're developing a method to even the scale, so to speak.
Excellent.
That's interesting. Yeah, I guess I never really thought about the cancer cells are already doing that, just like for our detriment, right? And I think when I first heard of Asgard, my initial like phrase was transforming cancer cells into snitches.
Yeah.
Right. So I guess it kind of works, but turning them into allies is a little bit better. It's more broad, I guess, because they can do all kinds of different things. And I guess I never really considered the idea that like, yeah, the tumor is already doing that.
Yeah.
And we're just kind of like using that same concept against them now. So
exactly.
Yeah, really, really cool.
Love it. Okay, we do have a question from Shifu999. Asgard Therapeutics is doing reprogramming cells as well. What's the main difference with this company, and what are the commonalities? So yeah, how are you guys different? How are you guys the same? And I guess the more important thing being the differentiator.
Yeah, no, absolutely. I'm very familiar with Asgard. I think they've done a very good job as to proving the concept of tumor reprogramming and having a proof of concept. They have a number of papers published on that end. With that said, our approach is different both on the biology, what we're trying to accomplish, and also in the actual therapeutic modality, right? So I believe Asgard is using adenovirus for their lead program. We're using mRNA. So that's one of the big differentiators on the modality side. And then on the like biology side, what Asgard is trying to do is replicate like a certain cell type, right? A conventional dendritic cell type 1. Mm-hmm. In our case, we're not necessarily trying to recreate one particular cell type. Instead, we're asking a different question: which functions we can activate within the tumor for maximum therapeutic benefit? Mm-hmm. And that involves antigen presentation, that involves immune cell recruitment, co-stimulation. So we're not necessarily going after a particular cell fate or try to recreate, that cell fate exactly. We're trying to turn tumor into the best ally we can have.
Yeah, that makes sense. So so on that note then, do you use a wide assortment of regulators, or is it like a very, very tightly designed assortment to do all those different things?
Yeah, so it's a combination of master regulators. We in the beginning, when we first started, we screened a bunch of them, and now we kind of narrowed down into top 3 that are different from Asgard's, and we're testing them in animal models right now.
Mm. And those 3 are able to on their own kind of perform all those different functions, or is it just you get this like natural array?
Yeah, so essentially we're trying to kind of activate different functions in the same cancer cell. Mm-hmm. And what we've seen so far in vitro and both in vivo is that it works across a variety of cancer types, which is very important for us. We don't want to change a composition of our drug based on the cancer subtype. So wide applicability is important. And then efficacy in vivo in syngeneic mouse models. That's something that we see. And then most importantly is the immunity or the resistance to cancer rechallenge. And that's what we observe as well. So I think it's basically a combination of different functions that we try to activate for maximum therapeutic benefit.
Yeah, I like it.
Nice. Cool. So yeah, can you walk us through just like some of the main results you've seen so far?
Yeah, so we've tested our drug in a CRC mouse model, and we see in vitro that we can consistently upregulate the pathways that we think are important for anti-tumor response and immunity. And then in vivo, since we have both the tumor, but also we have a functional immune system of an animal, you can think of our drug as kind of a match, right? Right now, the immune system is gasoline and is just like looking for the right spark to recognize the tumor, attack it, and then become resistant to it. So what we've seen in vivo is that we can have a complete eradication of the primary tumor site. And then on top of that, the surviving mice have resistance to rechallenge. So when we inject additional cancer cells into those mice, they don't grow anymore. So that's what we're trying now to replicate in additional tumor models as well.
And was not that like the dosing would translate directly to from mouse to humans, but is it a one-shot thing, or what is it?
So right now we're doing a series of doses. We did 3 doses for our in vivo study. Main reasoning is that mRNA is transient, so you might need to have a sustained expression or to achieve high efficiency. However, there are different mRNA modalities, as you know, that can also maintain that after a single shot. So we're actively exploring all the different tweaks that we can do to our drug.
Yeah, and I guess it would be like maybe superfluous to the therapeutic aspect of this, but it would be very good for like biologically to figure out if you were to give like the transient RNA, right? You transform these cells, you reprogram these cells into whatever it is they become.
Yeah.
Do they resort back to being cancerous again, or is there some kind of like epigenetic like imprinting that happens that would help with like the dosing, basically?
Yeah. So typically, you can think of reprogramming as sort of a domino effect, right? You just need like a one push for the master regulators to activate the pathways to initiate the cell reprogramming. But also, a lot of these master regulators auto-regulate themselves. So once you have the protein so it can turn on the endogenous gene, and that just stays on. And that has been shown across a variety of reprogramming methods. Like most notorious is NGN2 neuronal differentiation, where you just need a short period, a few days of ectopic NGN2 expression to activate the endogenous one, and then you don't need to add anything anymore.
That's interesting. I didn't know that the factors themselves will auto-regulate themselves.
Yeah,
that's cool.
It's quite common.
Yeah.
Walk me through like what drove that choice to use mRNA versus a viral vector?
Yeah, so I think looking at the clinical landscape, they were already clinical trials of intratumoral injections of mRNA by Moderna. And I know you guys already discussed some of the most recent Moderna news, which is very timely, because I was looking forward to that readout. I'm glad that it was. Yeah, I like that. I think there are different ways to assess a therapeutic modality, right? Like persistence, manufacturing, scalability, and then also the cost. And when you take everything into account, for us, mRNA made the most sense.
Yeah. Huh.
Awesome. Super cool. Okay. Yeah. So I guess, folks, if you have any questions for Rustam, by all means, feel free to make them known. I'm sure he would be glad to dive into any of the nitty gritty details that you're interested in, aside from stuff that's proprietary. Um, yeah. So like kind of walk us through like for that lead indication for that lead type of cancer you're going after. What drove it? Was it primarily like unmet need? Was that kind of where you've seen the strongest data so far? Why that one in particular?
Yeah, so unmet need is obviously a biggest one. Also, resistance to existing immunotherapy, like checkpoint inhibitors. And if you look at the colon cancer, it's actually the cases are on the rise. They're on the rise also among younger individuals. And a lot of times when it's diagnosed, it's already metastatic because younger people don't do colonoscopies. So that was there's also very high incidence of resistance to checkpoint inhibitors. And that made the most sense. Since our delivery is also intratumoral, we need to consider that for the lead indication. We need to consider that. And for colon, what happens is a lot of the metastasis goes in the liver. And even after resection, you might have some lesions that are accessible to administer a drug intratumorally. So taking everything into account, we decided to go with that.
So, okay. Then the idea is that the initial reaction can happen inside of the main tumor, and then you get the abscopal effect with like the immune response effectively targeting the metastases. I guess it's a little bit of a paradox. Like I get the idea of we're going after ICI immune or resistant tumors, right? But because you want to like target the unmet need, right?
Yeah.
But in my head, I think, well, wouldn't those tumors be relatively resistant to like this as well? Because they're very immunosuppressive.
Yeah, I mean, one of the things that we're trying to do is make them turn cold tumors into hot ones, right? And one way you can do that is to stimulate immune infiltration, right, at that particular site. So I understand it's not an easy thing to do. Maybe kind of a lower bar, lower risk would be to go after immune hot indication.
Sure.
But for us, we really feel like this kind of approach is what's needed to turn a cold tumor into a hot one. And this can be done also in combination with existing immunotherapies, right? To make them, because there's a fraction of patients that respond to immunotherapy, right?
Right.
And we're trying to understand why. So I think it could be quite complementary in that sense.
Yeah. Is there plans to do that, like with Keytruda?
Yeah, I wouldn't mention one specific drug, but yes, plans is
whatever.
To investigate synergy.
Sure.
Nice.
Yeah.
Just go shopping around with all those poor folks that are approaching their patent cliffs, and they need to find $25 billion somewhere else. Nice. Right on. Excellent. So yeah, so walk us through based off of what you've seen so far, what are the next steps for the company? What should we be looking for next that you can share without signing an NDA away?
Yeah. So obviously the ultimate goal is to test our therapeutic in humans. So we're actively charting path to that first in human trial. There are certain hurdles that we need to overcome before doing so. Namely IND enabling studies, as I'm sure you're aware. Some CMC work to ensure that we can scale and manufacture this drug at the capacity that's needed. And then having also conversations with regulatory authorities to understand the best path forward. So that's our goal for the upcoming year. With that, we're also, apart from lead indication, we're developing our cell codex platform. Because if you think about it, reprogramming could be applied to a variety of different indications too. Imagine if you had a way to make the right cell type at the right place for a therapeutic purpose. So we are working on developing better ways to predict these reprogramming factors through our platform. So we're not one asset company. We're actively thinking about the future and additional indications beyond oncology as well.
Interesting. Okay. So maybe something kind of along the lines of, well, we think of turning cancer cells to allies, but it's like, can we turn cooks into dishwashers? Different roles inside of the same kitchen.
Exactly. I think it's possible, and I think I've done it already in a lab, right? In vitro, this has been done many times. It's just choosing the right application in vivo for as a therapy. But I think everything is kind of converging. Also, the delivery part, which is very important. There are better ways to deliver genetic material. You can think about AAVs, different tropisms, and also mRNA based.
So my brain is now it's flying. What other indications could you do? Like, what would you? I don't want to like make you say anything you don't want to, but give me an example of a potential indication where, besides cancer, where this would be applicable.
So I can tell you what has already been done in vivo by others for different indications. Right?
Sure.
So,
okay. Yeah.
Parkinson's disease. You could technically convert reactive astrocytes into dopaminergic neurons right inside the brain, and that has been done in vivo in a mouse model. I think there are applications in ophthalmology where you can transform cells in the eye into degenerating retinal cells. There's application in diabetes type 1 where you can transform alpha islet cells into beta islet cells in the pancreas. So there are quite a few. The challenge there is delivery and avoiding off-target effects.
I love how I'm sitting here like, I can't think of one. And then you rattle off like 17. That's great.
I've been thinking about it for more than a year. So,
well, I guess my thing was like, wouldn't you, because in cancer, you're transforming the already pathological cells into cells that are useful. But anywhere else, you would be transforming like already useful cells into something else, losing those useful cells. But I guess like if you have the microglia in the brain or your astrocytes in the brain, if they're inflammatory, you can just transform them from not being inflammatory to being anti-inflammatory or dopaminergic, right?
Yeah. So you hit the nail in the head. It's basically you need to find a way to transform bad guys into good guys. Right?
Yeah.
And in the case of Parkinson's, that's a perfect example. You have those reactive astrocytes that are actually amplifying the disease pathology. So you're basically hitting two birds with one stone, right? First eliminating them and then making a beneficial cell type.
Right.
That's fantastic.
Yeah, that's cool.
Okay, we do have several more comments to get through. Folks, if you have anything else that you'd like to ask, feel free to sound off in the chat. From Shifu999, are there any scientific publications for reprogrammed bio out yet?
Yeah, not currently, but as at the right time, we're gonna publish.
Nice, excellent. And then another question, also from Shifu999, doing some great work in the comments here. Could this whole application also be applicable to sarcomas?
Theoretically, yes, but that's not something that we're investigating actively.
Gotcha. Okay. Well,
any solid tumor in reality, really, right?
Yes. As long as it's accessible for intratumoral injection.
Right. So has that been a limitation? I see you guys raised your seed back on my birthday, actually, May 27th.
Oh, really?
Yeah. So congrats on that. Yeah. I gave you my birthday present. Has there been any kind of like pushback? Because I think we were talking about oncolytic viruses earlier, and one of the biggest problems with oncolytic viruses is the requirement to do primary tumor injections. Mm-hmm. You guys had any kind of pushback on that? Like, how has the reception been to that limitation?
Yeah, when you mention intratumoral injections, that obviously comes up. I think what others like Moderna did quite well is that they showed that this can be applied to a variety of, like, I would say over 70% of solid tumors.
Oh, wow.
Obviously, you cannot go to GBM or something in the brain, which could be challenging. But we're also thinking about the next generation of how we can develop a therapeutic that can be administered IV. And go to that. That's obviously a higher technical bar. That's something that we're thinking at the company as well.
I wouldn't write off the brain tumors. We've got a couple of companies who are doing intratumoral injections of gliomas, like Siren Bio or Trogenics, and they seem to be having some success. So
yeah. I'll check them out.
Yeah.
Siren Bio is super cool. I was there the day that they launched at ASGCT back in 2023. And then their CEO Nicole Polk has like limitless energy. I don't know.
Yes, I think they are in the same.
Now you mentioned energy. Oh, I know that lady actually.
No, I think they're in the same like bio incubator as we are. We're at MBC Biolabs in the Bay Area.
Wow.
I think they're also maybe at a different location, but yes.
Right on.
Yeah.
Well,
if you're within a mile radius of Nicole Polk, you probably know.
You'll feel it. Yeah.
There's just a blur in a red dress. Right on. Anyways. Excellent. Shout out Nicole, friend of the show. She was our very first guest on the Biotech Voyager when we launched it.
She was.
Yeah, she's great. Cool. All right, excellent. Well, I know we've only got a couple minutes left here with you, Rustam. We really appreciate your time. Folks, if you have a question, now is the time. We're coming down to the wire here. But there is one question we just always like to try to ask at the end to each one of our guests. It can be a conglomeration of everything we've talked about so far. Ultimately, we hope to just kind of make a nice little soundbite clip from it. The goal being, and the question is, like, what's slept on in biotech right now that's going to be a big deal in a couple of years?
Reprogramming. Application of reprogramming.
Everyone always does this.
Exactly. That's all I think about, right? But I think if you look at the evolution of therapeutic modalities, right? So first we started with drugs and then ultimately biologics to target one gene. Then you have cell therapies where you can take cells from the patient, modify them, put them back. Now you have in vivo CAR T that you actually don't need to remove cells from a patient. You can just modify them inside the body. But what if you can make the right cell type, the right cell state in the right place in the body? And that's what reprogramming does.
It is kind of like the natural extension and like the evolution really of like the in vivo CAR concept. Because you're transforming T cells into CAR T cells. It's reprogramming them. But this is like true reprogramming. And it's in vivo genetic medicine. Like it should, I guess that's with the attraction of in vivo CAR T, knowing that this is like almost like the bigger brother of in vivo CAR T. Like
I think we like with reprogramming therapeutics, we are where in vivo CAR T was maybe 5-10 years ago.
Okay.
Where people like didn't believe, right? Is it really possible? Can you even, edit T cells selectively in the body? And now we have clinical validation from multiple companies. So I think reprogramming will be there in a few years.
Fantastic. Excellent.
Exciting.
Well, for any investors who are in the audience, it seems like a great candidate for some additional investment in the future. So definitely really excited by the approach that you guys are taking, and it's great to see it's been validated not only with you guys but also in other companies as well. Like it's a growing, emerging space. So exciting to see, especially as it kind of looks like we're coming out of the bottom of the biotech trough, right? We're getting a lot more IPO activity, a lot more mergers and acquisitions for those later stage assets, which is ultimately going to be a trickle down effect into the phase 2 and phase 1 and preclinical companies. So really excited to see where things go over the next couple of years, and we will be following you with interest. So thank you so much for your time, Rustam.
Yeah, awesome guys. Thank you.
Thanks so much. Cheers. Right on. That's so cool.
Super cool.
Yeah.
So I'm surprised that it's not being done more often, but I guess like knowing how to do it. He mentioned he's been in reprogramming for a long time. You've got to be like really careful and like really know the factors that you got to use, and like it's probably such a balancing act. Especially knowing that it's like self-perpetuating almost. If you touch the wrong button, that could be problems. But so you gotta have like a lot of experience, know what you're doing. There you go.
Yes, we agree.
Oh, Shri. What's up, dude?
Yeah, thanks for coming.
Yeah. So, what do you say, Ben?
Yeah, no, man, this is I'd say we're just about rounded out here. Folks, if you're not yet subscribed to the Biotech Voyager on YouTube, if you can do us a big favor and leave us an additional subscriber in yourself, that would be fantastic. Our goal really is, again, to map out the early stage signals. Follow us on Instagram or on Twitter. I see that we got a couple people coming in from X today, so that's awesome to see as well. So thank you to everyone who has joined the show today. If you would like to do all of this by yourself without us, you can go to thebiotechvoyager.com. You can create a free account. It's all free. Nothing on there costs you anything, and you can go do all of your own assessments of the early stage biotech space. It's a really comprehensive tool that we just want to provide for everybody inside of the space. Hundreds of you have signed up for it already, so
almost 500 so far. It's been about 2 months, which is great.
Yeah, we're about 2 months in. And then, yeah, I guess the other thing would be the fundraising and BD engine for biotech. We've created something very specific for folks in that area, a comprehensive tool that's able to just do an incredible amount of work that's built by biotech people for biotech people.
It was built by me.
It's
HubSpot on steroids who's obsessed with biotech and research. That's basically how you can think about it.
HubSpot on all sorts of next generation.
Mind-altering therapeutics.
Yes, that's right.
Next gen neuropsychosis drugs.
HubSpot on longevity protocols. Mm-hmm. Excellent. Well, folks, we'll probably leave it there today. Thank you so much for coming. As always, my name is Ben. I'm joined by...
Don't say it.
I'm going to do it.
My name is Jeff.
My name is Jeff.
But yeah, folks, we'll leave it there. Thank you so much for coming. We'll be back on Tuesday. Also, stay tuned. In the coming weeks and months, we have plans to do live conference coverage, so we might be able to see some of you in person. But additionally, another component of something that we're planning on building out is going to be debates. It's one thing for me and Jeff to kind of come on and talk to one person. We want to start facilitating some of these big questions in the space around different kinds of events and different kinds of debates that we should do. So probably one of the first ones that we'll do will be lentivirus versus LNPs. So stay tuned for that. Ooh. If you have any suggestions for other types of debates that we should do between different modalities or different approaches, do let us know. And then finally, the other thing we'll be doing as well is potentially doing like thematic episodes. And so thematic episodes being something like, we've seen a lot of interest recently inside of like these active immunotherapy space. We are in talks potentially to do like a dedicated episode to that where we'll bring on a bunch of different experts and kind of interview them sequentially to learn more about this really exciting emerging area of biotech. So if you have any thoughts or perspectives on all of that, please do let us know one way or another. You can find us on LinkedIn. We're chronically on there. But that's about it for today. All right. Thank you so much, everyone, for coming, and we will see you next time.
Bye. Peace.